Yes, once again, grade 12, paper 2, section A. Let's work it together right now, right here. Let's just check some technical questions that are here right now. You can see we are going to talk about the structure of nucleotides, the building blocks of DNA. We are going to look at this, and then I'm sure that you know the structure of nucleotides. How do they work? What do they do? We are going to talk about the phylogenetic tree diagram. We are going to talk about complete and forms of dominance, complete and incomplete dominance. I'm sure you know that codominance. You know this. What is it? How does it come? We talk about the terminologies that are quite common and always there in paper 2. You can see all these are quite good long terms that are very important here. We define them together. We are going to talk about cell division, meiotic cell division. As you can see, we're gonna work them together right now, right here. Just be patient, follow up, and let's work it together as a class. Please don't go away, keep connected. Without wasting any time, we wanna go and work out the exact questions as they are. So we are going to work these questions, section A. This is a September paper 1 paper, which is from Eastern Cape, and we want to work it right now. So what is happening here is that we are given multiple choice questions that are here. Uh, which of the following statements best describes a difference between a chromosome and a gene? A chromosome, a gene. I'm sure remember the basic terminology in genetics. What are chromosomes? Protein-like structures that are responsible for carrying genes, which are responsible for inheritance of characteristics, basically because they carry the genes. Genes are attached on chromosomes. So what is the what describe the main difference between the two? A gene is one section of a chromosome, and a chromosome may carry many genes. uh, I'm sure, let's just say that we are saying the main difference is a gene is a section of a chromosome. That is true. And chromosomes may carry many genes. And that is actually correct. And then a chromosome and a gene are identical. Not a gene contains the entire DNA of a human, while a gene is portion of a DNA. A gene is larger than a chromosome. So what we can see is that a gene is a section of a chromosome, and a chromosome may carry many genes. That is why we talk about the various characteristics that we have in organisms, which are passed on because they are carried by chromosomes. So these characteristics, there are quite a lot of them. I'm sure I remember we talk about a human being having 23 pairs of chromosomes, which are responsible for various characteristics. And I'm sure that we can agree and we can work it out together there. In order to make the following nucleotide a monomer of DNA, replace dash. In order to make this one a monomer of DNA, what has to be there? So we have got phosphate, we have deoxyribose sugar, but there is a problem there where we have uracil. We are saying uracil does not exist in what? In DNA. This is uracil. It is found in what? In RNA. So we are saying this is where the error is. So we have to remove uracil. So the answer should be saying we remove uracil, and what should we put there? A DNA. I mean a nitrogen base that is in DNA, which is which is replaced by uracil is what? In RNA it is what? It is thymine. Thymine, it is found in what? In DNA, and RNA contains what? It contains uracil. So uracil, we have to do replace uracil with thymine. Uracil must be replaced with thymine. That is what has to be done there. So sometimes you'll be given this question And you need to be able to identify what is this. This is a nucleotide, a building block of this is a building block of DNA, RNA, uh, in terms of their structural representation. So please don't forget, don't make mistakes. And you can see there we have got the phosphate, we have got the deoxyribose sugar, which is in terms of what? In terms of DNA. In terms of DNA, if it was RNA, it's what? It is the ribose sugar. If it is RNA, the sugar is what is ribose. So we are saying DNA, it has got deoxyribose sugar. That is one thing that we need to take note of. Remember, we are just comprehensively uh, revising right now. Scientists recovered the body of a whole mammoth from the frozen soil of Siberia. The DNA sequence of the whole mammoth was very similar to DNA sequence of the African elephant. Which of the conclusion is best supported by this information? So what we need to understand is that there is a relationship between these two. The woolly moth, which was discovered in Siberia, is also having a relationship with DNA sequence, which is found, I mean, the DNA sequence of the two in African continent versus the Siberia, they are the same. So, I mean, they are related. So there must be a relationship in terms of their evolution. So now, the African elephant evolved directly from the woolly mammoth. The woolly mammoth and the African elephant have a common ancestor where they also which they both originated from. They have a common ancestor where they all come from, so that they share their DNA. The DNA is closely related, hence they are able to be linked together. Lamarck's theory of evolution was rejected because he incorrectly proposed that in terms of this, I remember we said Lamarck versus what? Versus Charles Darwin. These are the two main outspoken theorists in terms of evolution, Darwin and Lamarck, where Lamarck's theory was rejected and substance and acceptance was given to Darwin's theory. So now we are saying the the theory of Lamarck was rejected because he correctly proposed what, according to his principles. I'm sure that we have already also discussed it in what in our other videos, theories of evolution. Please check also on channel theories of evolution. Theories of evolution, they are there, well explained and also analyzed. So the Lamarck theory was rejected because acquired characteristics are not inherited. We said that acquired characteristics are not inherited. These are characteristics an organism gets through its lifetime. So they are not inborn. They are not genetically influenced. So acquired characteristics are not inborn. I mean, are not inherited. That is the major reason why the theory was rejected. And then variation, which derives natural selection, is not caused by which derives natural selection, variation. Other times we want to see sources of variation class. Sources of variation, we have got quite a lot of them in terms of, say, now we said meiosis, which is because of crossing over, which is caused because of also random assortment of chromosomes. We also talk about mutation. We also talk about random fertilization is also other forms of what of sources of evolution. So number 5 is saying variation which derives natural selection is not caused by what? Variation which derives natural selection is not caused by what? Is it mutation? Can it be phosphorylation, chance fertilization, random arrangement of chromosome during meiosis? So if you can see, variation which derives natural selection is not caused by phosphorylation. Phosphorylation cannot cause, we are saying is the one that cannot apply in this case. Mutation can do, chance fertilization can do, random arrangement of chromosomes during meiosis, it as well can work. One of the following is not an example of reproductive isolation. We are talking about speciation. We talk about reproductive isolation and also geographic isolation, which results in formation of new species. So, which of the following is not an example of reproductive isolation mechanism that prevents two species from producing viable fertile offsprings, like to be able to what to reproduce correctly or to reproduce a normal offspring? Which one of that cannot can is not an example? Habitat isolation, it can do. Habitat isolation, we are saying habitat isolation organisms can be can be can be separated because of habitat, is not an example of reproductive isolation. So we are saying organisms are not able to reproduce because of being separated. So which of these can mechanisms, which of one of the following is not an example of reproductive isolation mechanism that prevents two species from producing viable offsprings? So we are saying which one of that is not an example? Habitat isolation, we say it can do. Behavior isolation, we can put a question mark on that. Hybrid sterility, we are saying it cannot cause viable offspring. It cannot cause viable offspring. Mechanical isolation also, we are saying, is not an example of reproductive isolation. So now, mechanical isolation, it can also result, habitat isolation can also cause hybrid sterility. It obviously means that the offspring is what is not fertile. So behavior isolation, it cannot cause, it cannot be, is not an example of reproductive isolation, which we say that there is also artificial methods which people can, where people can bring organisms in contact, and then they can be able to reproduce and make a viable offspring because of artificial or selective breeding, which can also take place. So our answer is B. uh, the pedigree diagram below shows the inheritance of homeophilia in a family uh, where dominant H is normal blood, and it also is also a dominant gene, and recessive H, recessive H is recessive gene causing hemophilia. So the disease is a sex linked that is caused by a recessive allele. It is caused by a recessive allele. In this case, the recessive allele is the one that is causing that. What is the genotype of a family member? Number three. What is the genotype of a family member? Number three. A family member, number three. What is the genotype of this family member? Number three. So I said that when we have got situations such as these, we must be able to understand that when the circle or the individual is unshaded, unshaded means non-homophilic. But however, we have got an affected son here, a son who is affected. How do we have an affected son? The son inherited a recessive disease causing allele, disease causing allele from the mother. That is why the son is affected. So if in this case the mother has got this condition, it means this mother, she is supposed to be what? She is supposed to be a carrier. So a carrier mother must have, a mother who is a carrier, she must have what? She must have a recessive and a dominant allele, where we have got our XX and then our small h, as that is supposed to be a heterozygous she must have a heterozygous what? She must be a heterozygous expression, which is something that has got the dominant and recessive age. That means is A. B is actually normal. B, this one, it means the person, the mother is normal, and then she cannot be able to give an affected son. And also, according to number C, it is wrong because this mother, she is totally affected. But according to the pedigree diagram, according to our diagram there, it is showing that the mother cannot be affected because it is unshaded. It is unshaded, which means that the mother cannot be in this case. And again, D can be is also correct, is also wrong because it represents a male instead of a female. Instead of a female. Then we are saying the correct answer is what? The correct answer is what is A. The advantage of cloning is that advantage of cloning is that it is the advantage of cloning is that it is will reduce the variation within a population. Therefore, it cannot be an advantage. If it reduces, it cannot be an advantage. Produce genetically identical organisms with desirable characteristics. That is exactly what cloning does. Cloning produces identical organisms with desirable, with much needed characteristics, with much required characteristics by men. So therefore, it is an advantage. Uh, that is the advantage that we can basically talk about and which we can actually give in this regard. 1, 2, 3, and 4 in the diagram below represents a pair of chromosomes. The correct interpretation of the diagram is that what is the correct interpretation of the diagram? The correct interpretation of the diagram, we have got the first here, we have got a homozygous. We have got a homozygous, and here we have got a heterozygous expression that we are seeing there. And then what is the question asking? Is saying the correct interpretation of the diagram is that what is the correct what is the interpretation of the diagram? The correct what is the interpretation of the diagram? The question seems like is incomplete. Unfortunately, I don't it was not well copied, but anyway, we are saying it is incomplete in this regard. The question does not show the whole sequence of what is supposed to be asked in this respect. Uh, an error happened in terms of pasting it. So let's go to the next question. A cross between a white cat and a black cat produces a white cat with black patches. It is a cross between a white and a black, and then it produces, this is the offspring. The offspring, it has got, it is a cat with some black and white patches. So what is this form of what? Of expression. This illustrates what we are saying when these two are crossed together, we have got this one. So black and white are crossed together. We have got our white. It's crossed with what? With black. And then we have got white black patched white black. We have got a white black cat as what? As the offspring. So it means this is what? This is a codominance. This is a codominance because both of the colors are what? Are being shown in this. We are seeing the colors are almost equally expressed. When we say codominance, it means both colors, white and black, are equally expressed in this young or in this offspring of this cat. So give the correct biological term for each of the following. formation of two or more species as a result of separation of the original species into different groups by geographic barrier. It is called what? We say that that is called geographic speciation. It's called geographical, it's geographical speciation, or it is also called allopathic speciation. It is also called allopathic speciation, uh, the process that enables organisms with different desirable characteristics to survive and reproduce, whereas organisms with undesirable, they die. We call that it's a definition of natural selection. Ones which have got the desirable characteristics, they survive. Those without, they die. We call that natural selection. Natural selection. And then it is by nature itself. There is no human interference. Hence, it is natural selection. Chromosomes that determine the sex of an individual, we call them what? We call them sex chromosomes. These are called sex chromosomes, or we call them what? Gonosomes. Gonosomes or sex chromosomes. They are the sex chromosomes that defines the gender, the sex of an individual. The physical appearance of an organism, we call that the phenotype. The physical appearance or the outward appearance of an organism, it is called the phenotype. The inward appearance that is controlled by the genes inside, we call that genotype. But the physical is the phenotype. They are the one that we can see. A physical, a process that involves the insertion of a diploid nucleus from an embryonic somatic cell into an unfertilized egg, which then becomes implanted into the uterine wall. We call that cloning. This is a cloning process. Cloning, I'm sure if you can also refer to channel, there is also a diagram, a video. Refer to channel also for a video which also explains the cloning process, reproductive cloning. How is it done? Uh, the point at which the chromatids of a homologous chromosomes cross over during the first prophase of meiosis, we call that what? The point of intersection. We call that chiasma. It is called chiasma. We call that chiasma, and then an inheritance in which a particular trait is produced by the interaction of many genes. That is called polygenic inheritance. Many means genic, polygenic, polygenic inheritance. Polygenic inheritance is what you call that process. It indicates whether it's A, B, or none, different forms of a gene which occur at the same locus, the position of a gene on a chromosome, a pair of homologous. What we call that, it is what? It is an allele. An allele is called a variation of a gene, which we are saying it occurs on the same locus on a gene. In anticodon, it is something to do with the protein synthesis process from the what? From the tRNA. And then the process by which animals or plants are selected and allowed to interbreed with selected animals or plants to produce desirable. When it is selected, obviously, when there is selection, it means there is human interference. Therefore, it is artificial. What is selected is allowed to breed, so it is artificial selection. Natural is when it happens on their own without human interference and the desirable characteristics of allowing survival. The number and shape arrangement of all the chromosomes in the nucleus of a somatic cell. We are saying the arrangement, the number and shape. We say that shape number equal that we call that the karyotype. The karyotype, the genome. We are talking about the total number of genes within a population species, but the arrangement and structure of chromosomes, we call that the karyotype. The process by which DNA makes copies of itself. What is that? When a DNA makes a copy of itself, we call that what? DNA replication. It is called what? DNA replication. Replication is when a DNA makes a copy of itself, but in this case, it is therefore non-point mutation and frame shift. These are forms of mutations which does not result in formation of a DNA copy. The region at which chromatids are held together, chromatids are held together, the region at which chromatids are held together, that is, they are held together at the what? At the centromere. The centromere holds the chromatids to form it a chromosome. They are held together by the what? By the centromere. Number 6, a cell that has only one set of chromosomes, we call that a haploid. When it has got one set, it's called haploid. This one, it is when it has got two sets. So one set is a haploid, and then a specific pattern of bands that represents a unique sequence of nucleotides that resemble a barcode. We call that DNA what? Profiling. It's called DNA profiling. DNA replication, we're saying it's making a copy of itself. Making a copy of itself. Making a copy of itself is what we call that. Is what we call this. So now I'm sure you can see and you can be able to understand this. It's also just an understanding of definition of terms that you are being tested in this case. The diagram represents meiotic division of two cells. Both A and B undergo certain unusual phenomenon and gave rise to a male and female gametes respectively, A and B. So male and female gametes are given there. And then the question is saying, identify the phenomenon depicted by cell A and cell B. What is the phenomenon represented by A and B? What we call that is called non-separation, non-separation of chromosomes, chromosomal non-separation, non-disjunction of chromosomes. We call that non-separation or disjunction of chromosomes. And then in which organisms does this phenomenon usually occur? We usually have it occurring in what? In plants. It normally occurs in plants where we have got conditions such as this. And then what is the phenomenon generally known as? It is known as what? As we normally call that polyploid. I'm sure you have seen the advantages of polyploid in agriculture. Polyploid. Polyploid, we say it is called polyploid, which we are saying advantage of polyploid in agriculture, producing organisms that are bigger, organisms that are just totally different from the normal ones. And then what would the chromosomes number be in male gamete, female gamete, and that? So according to this, we are seeing that there is non-separation here, so it remains diploid. As you can see, it went down as it is. We have got 1, 2, 3, 4, 5, 6, 7, 8, which has got 8. We have got 8, 8, 8. We can say it actually represents that it it has got 8 gametes, and also we have got 8 in what, in the female there. So we have got the total number of male gametes. They did not separate. We have got 80, which we can say that it is actually a full set, or we can say it is diploid, or we can say also it is what, it is 2n. So as you can see, we are saying the chromosomes did not separate. So we have got all the full set as it is. 2, 3, 4 times 2, 3, 4. So we have got 8 here. We also have 8 there. So it actually remains the same, and then they keep on, they are up, they are taken into, we say they form a zygote, a gamete here, which is having the total number. This total was adopted. It has the total here of the parent, total of the parent cell. It has total of the parent cell. The parent cell has 80, and then also this, the daughter cell, which is the gamete, also has what? It has also the total number, which is what? Which is 80, and then it also has 80 there. So the number 80 remains there. We also have the same answer. It has got 80. It is diploid. It is also 2n. Any of that can be correct. And then the zygote, obviously, what is happening with the zygote is 8 times 2. The zygote is, this zygote is equals to 8 times 2, which gives us what? It gives us 16. It is giving us 16. It's 8 times 2, which have got, which can give us 16, which can give us 16. Remember, 16 can also be represented by what? By 4n, which we said n represent the number of chromosomes. So it is 4 times 4, which is going to give us what? 16. And the answers are what? Are correct. I'm sure you remember that, to say if the same thing as to say 2n when it is a normal gamete. If we say 2n, 2n is equals to, which we said that 2n represents the number of chromosomes, whereby we say that it is what? It is diploid. It is diploid. So in humans, when it is that 2n, which is 2n, where n is equals to 23, and then we now have 46 if it is in humans. If it is in humans, therefore we can get it like that. The same applies here to say that our n, our total number of chromosomes are there, which we say that is 8 times 2, which is an addition of a male and female gamete coming together by fusion. What was the chromosome number shown in the diagram above of cell A and B before meiotic division took place? Before meiotic division took place, we are saying they are actually given. We said we already counted that. I'm sure this is answered before meiotic division, because meiotic division was supposed to come here where we have the gamete. This is the stage of meiotic division. Meiotic division was supposed to be shown here, but it went down as it is, where we're saying that the first number was 80, and therefore the number before meiotic division in A and B, it was what? It was 80, which caused us to have 8 times 2. State 2 ways in which the phenomenon shown above is important to plant breeders, which I already have mentioned there, that in agriculture, it results in larger plants are produced. It results in uh, uh, larger fruits. We can talk about also more yields. We can also talk about more money or income or profits when such large fruits and whatever are sold. You understand? Those are the advantages that we need to take note of. Those are the advantages that we can talk about. So basically, what we need to know is that polyploid in agriculture, advantages of polyploid are like those ones: more yields, more money, more fruits or larger fruits, and then something, everything to do with an increase in size, because this is a polyploid resulting from non-separation or non-disjunction in plants during flower formation or during the formation of gametes. Then that is the reason why we have such. So class, thank you very much for your time, and we are going to meet again in section B, which we are going to look at some other basic technical questions that we need to know as we go to write exams. I think as you go tomorrow, as you go next week, as you go to November, December, all things will be well. Let's keep connected and keep following. Quite a lot is going to happen on this channel. Refer friends, refer brothers and sisters, bring them to the right channel, the right channel to take them far. Uh, Thank you very much once again. Until we meet again tomorrow, keep following, turn on your notification buttons so then you won't be left out.